Welcome to today's lecture on cell communication and immune response. In this session, we will dive into the fascinating world of cell signaling and the complex mechanisms of the immune system. Our exploration will cover various topics, ranging from G-protein coupled receptors, GPCRs, to the immunogenicity of therapeutic antibodies. So, let's begin our journey into the intricate world of cell biology. Cell communication is essential for maintaining homeostasis and coordinating various physiological processes. The immune system, one of the most intricate networks in the human body, relies heavily on cell signaling to combat pathogens and maintain overall health. Understanding the mechanisms behind cell communication and immune response is crucial for providing insights into diseases and developing targeted therapeutic interventions. GPCRs play a pivotal role in cell signaling, including immune response modulation. These receptors span the cell membrane and transmit signals upon ligand binding. GPCR activation triggers a cascade of intracellular events, including the activation of G proteins and subsequent stimulation of various signaling pathways. Examples of GPCRs involved in immune response regulation include chemokine receptors and adrenergic receptors. Toll-like receptors are a family of membrane proteins that recognize specific pathogen-associated molecular patterns, PAMPs, and activate immune responses. TLRs are crucial players in initiating inflammation and promoting the production of pro-inflammatory cytokines. Their activation serves as an alarm system, alerting the immune system to the presence of potential threats, such as bacteria or viruses. Apoptosis, or programmed cell death, is a highly regulated process crucial for maintaining tissue homeostasis and eliminating damaged or infected cells. Multiple signaling pathways orchestrate apoptosis, involving key players such as caspases, BCL2 family proteins, and death receptors. Imbalances in apoptosis signaling can contribute to various diseases, including cancer and autoimmune disorders. Cytotoxic T lymphocytes, CTLs, play a vital role in the immune response against infected or cancerous cells. These specialized immune cells can recognize and destroy target cells presenting antigens on their surface. CTLs employ a unique arsenal of effector molecules, such as perforin and granzymes, to induce target cell death, ensuring the elimination of harmful cells and maintaining immune surveillance. T-cell receptors, TCRs, are responsible for recognizing antigenic peptides presented by major histocompatibility complex, MHC, molecules. The human body produces an astonishing diversity of TCRs to ensure the recognition of a wide range of pathogens. This diversity is achieved through genetic recombination and somatic hypermutation, enabling T cells to respond efficiently and specifically to foreign invaders. B-cell receptors, BCRs, are antigen-specific surface receptors on B cells that recognize and bind to antigens. Upon antigen binding, BCRs initiate a signaling cascade that triggers B-cell activation and subsequent antibody production. BCR signaling involves complex interactions with co-receptors and downstream molecules, ensuring proper immune response activation and antibody-mediated defense mechanisms. Regulatory T cells, TREGs, are critical for maintaining immune tolerance and preventing excessive immune responses. TREGs suppress the activity of other immune cells, such as effector T cells, through various mechanisms, including the secretion of immunosuppressive cytokines and direct cell-to-cell contact. Dysfunction or deficiency in TREG function can lead to autoimmune diseases and chronic inflammation. Cytokines are molecular messengers that mediate immune cell communication, regulating inflammation, cell proliferation, and differentiation. These small proteins play a pivotal role in immune response modulation, coordinating interactions between immune cells. Examples of key cytokines involved in immune regulation include interleukins, tumor necrosis factor, TNF, and interferons. Antigen presentation is a crucial step in immune response initiation. Antigen-presenting cells, such as dendritic cells, engulf and process antigens, presenting antigenic peptides on their surface in conjunction with MHC molecules. Cross-presentation occurs when exogenous antigens, such as those derived from infected cells, are presented by MHC class I molecules, priming cytotoxic T cells for immune defense. Therapeutic antibodies have revolutionized the field of medicine, offering targeted therapies for various diseases, including cancer and autoimmune disorders. However, their immunogenicity, or the potential to induce immune responses, can limit their effectiveness and safety. Understanding the factors influencing the immunogenicity of therapeutic antibodies is essential for optimizing their design and clinical applications. Cellular senescence refers to the irreversible growth arrest of cells, often associated with aging or stress. Senescent cells can accumulate over time and impact immune surveillance, leading to inflammation and tissue dysfunction. Understanding the communication between senescent cells and the immune system is crucial for developing strategies to prevent age-related diseases and enhance immune surveillance. In conclusion, cell communication and the immune response are complex and orchestrated processes involving an array of signaling molecules, receptors, and immune cells. The topics we explored today shed light on various aspects of these intricate mechanisms, emphasizing the importance of understanding cell biology for medical interventions. By comprehending the details of cell signaling and immune regulation, we can strive towards developing novel therapeutic strategies and improving patient outcomes.